I think I'll go ahead and start with the title, of course, being Bridging the Gap Between Data Scientists, Clinician, and Biologists using the CRDC and the CGC Cloud resources. Now, this is something that is very close to my heart, and I think for those of you who were able to join the CRDC all hands meeting yesterday and the day before yesterday, so you probably had a lot of this information, but I wanted to start with acknowledging my terrific branch team members that could not do any of the work without their help. They're fantastic. They're great scientists and really great people to work with in general. Just a little bit about the actual branch. What we're doing is that we provide comprehensive and integrative data analysis for NGS and proteogenomics for the NCI intramural, extramural, as well as the NCI-initiated projects such as TCGA, previous projects like Targets, the pediatric form of the TCGA, and Apollo and CMAX. And don't worry about some of these acronyms. You know, when you're in the government, you have to kind of use these. If you're interested, I'll be happy to go into more detail with those. We also, in addition to the actual data analysis, we're very much interested. I'm very passionate about consulting and training, trying to disseminate the information or the knowledge that the team has to the rest of the NCI community. And while we're doing that in the process, we also have some tools that the branch team has actually developed within the house, and we're now trying to disseminate that as an open source. So with that, I think I wanted to, again, there is obviously link here for our branch. If anyone is interested, they can go and look at some of our publications as well as some of our expertise. And if there are any potential for collaboration, we'll be happy to, again, discuss that with you in the future settings. I think I borrowed this, since this is such a broad topic, I actually borrowed this slide from Eric Green. He gave this talk in 2001, where his dream was the 5 domain of genomic medicine or genomic research, where he was thinking about the structure of the DNA, obviously all the way to treating patients. So he basically called this the from bench to bedside. And there are, of course, quite a few steps to be able to get to the bedside. But I think that dream has now come to reality. And as you are aware, very well aware of the fact that with the help of the computational biology and molecular medicine, now there is an enormous amount of help in terms of early detection and genetic predisposition for different diseases, improved diagnostic, obviously, of the diseases because of this. Some of the tools and algorithms are available with the genomic medicine. The pharmacogenomics, of course, is a very, very huge field, trying to design actually customized drugs and using the personalized medicine, as I'm going to mention 1 or 2 of them in the next slide. So this is really something that can come to fruition. And I think more than anything else, this is an anecdotal example that I can bring into display here, is that 15 years ago, when you actually speak with most of the oncologists that are treating cancer patients, specifically lung cancer, they were literally working with basically 3 different types of cancer. They're adenocarcinoma, large cell carcinoma, and squamous. But fast forward to today, and this is an older slide that I actually got from, I think Ms. Sharpless was giving a talk that I actually got this from him. But anyways, the bottom line is that with the improvement of this potential molecular medicine and precision medicine, application of these bioinformatic tools and computational biology, now we have seen the heterogeneity of the non-small cell lung cancer. It is incredible how heterogeneous it is, and how, based on its molecular manifestation, there are different types of lung cancer. For example, the KRAS mutation, the EGFR mutation, the ALK, and many other ones. Of course, there's quite a few of them that we still don't know. And this potential molecular diagnostic now opened the door for enormous amount of fantastic treatments. As a matter of fact, we were involved in one of these research with the KRAS with the Frederick National Lab, and the KRAS, after 44 years of being obviously identified, the first diagnostic or the first basically treatment was administered about 1.5 to 2 years ago, Amgen being the pioneer in terms of potentially targeting the RAS. So the point is that this is really happening now. You know, we are really in the cusp of literally explosion of this genomic medicine, precision medicine. The issue is, and this is something that I'm again very passionate about, is that unfortunately the so-called training, or the way that we actually work with some of our collaborators, whether we are working from the data production, let's say within the laboratory, where the laboratory scientists actually producing the data, to handing this to the bioinformatician or computational biologist, and eventually that goes into a clinic and given to actual clinician to use that data. And the way I look at this is like a running relay, where the data is now being produced in the lab, and then the data is basically given to the bioinformaticians or computational biologists, and there is no communication or very little. So it's a linear approach. And I think there's a bit of a problem with that approach. And we wrote Barbara Dunn, my colleague, and I wrote a commentary in Cancer Research in 2019 addressing this issue, that this can be a problem, because now with this explosion of this new technology, the fact that there is no communication, this could potentially lead to some errors. And there are obviously a lot of examples, which we're not going to go over right now. But the goal here is that we have to have a new approach of actually communicating a little bit better. Why? Because generating all this data but not knowing how it was generated and where it was generated can be problematic. And what do I mean by that? There is actually a very big consortium, the CQC2. It was headed by Wiming Zhu. I think he gave a talk in the CGC Webinar Series as well a few maybe months ago, if I'm not mistaken. But this is a paper that we were part of. Working group, my branch was also involved in this, and here we are showing best practices in terms of doing NGS data production and data analysis. I'm just going to go over the slide very quickly. Here you're looking at potentially different time points for treating samples with FFPE, the formalin fixation, and as you can see, the global imbalance value, as it increases, that means that the rate of error is increased. So what you're seeing here is that the 1, 2, 6, 24 hours for whole genome and for whole exome. Again, I don't have to go through too much detail that the fixation time increases, the actual value, the GIV value increases too, which means that by fixing your tissue or the samples, you could actually introduce errors. And this is even more substantiated in the case of whole exome. So I guess the idea here is that if somebody is giving you some data, you ought to be aware of how they were actually produced and how old the data is, and knowing the lab work will help determine some of the artifact in the future analysis. And of course, this is another figure from one of those papers that was published in Nature Biotechnology. And here I'm just showing another potential error that can be introduced in the laboratory, where when you're doing sonication to actually prepare your DNA for your exome sequencing, if the sonication is done properly, let's say if you're fragmenting DNA within the range of about 280 base pair, that looks really good. But as the let's say sonication time increases and the actual fragmentation is now getting smaller than, let's say, around 180 base pair, now you are introducing oxidation in the process of sonication, which could actually introduce a conversion of a G to T and C to A. As you can see, as the actual fragment drops, the global imbalance value increases, specifically for those exome sequencing, for the whole exome sequencing. So the goal again here is, these are basically different sites that were doing this study. The goal here is that there has to be some standardization, some degree of communication among different domains. And I think my next slide sort of reiterate on that, that rather than having this linear approach where no one basically talk to each other, and the bioinformaticians and computational biologists work in silo, and of course the so-called bench scientists are working on their own, just producing the data and then don't worry about what happens next. We're basically suggesting or promoting this idea of a integrated type of work. And then at the minimum, there has to be a multi-domain expert or some way of form. There should be a circular way of actually communicating with one another. And that includes, of course, communicating with the bioinformatician has to be able to communicate with the bench scientist, and the bench scientists, of course, have to communicate with both the clinicians and the healthcare provider. As a matter of fact, this is already happening in some of the big cancer centers, such as Hopkins, Sloan Kettering. They have now team tumor boards, where in the tumor board they do contain, or they do invite computational biologists to sit in and give their input in terms of the identification of some of the mutations, whether it should be considered and how deleterious maybe, or some of the potential variant of unknowns. So there's a lot of stuff that again are coming up that I think we're learning that we should be inclusive rather than working in silo. So with that in mind, of course, this is a long-term, you have to train physicians to really learn about genomic medicine, about some of these potential technology. In the meantime, I think it is important to provide at least some degree of a platform for some of these basic scientists and physicians to be able to use to understand some of these. And most, as you know, better than I do this, that most of the algorithms and tools that are being used for genomic medicine are actually written in code command lines, and majority of the scientists, clinicians are not very savvy in terms of writing codes or reading code. So for that matter, I think CGC now provides a fantastic opportunity specifically within the CRDC in order to provide a specific, convert these potential codes or scripts into point and click and UI-based type of tools that can be utilized by some of these potential non-bioinformaticians. Now, again, I'm not going to dwell over too much about the fact that there are so many challenges, as you can imagine, with the amount of data that is obviously being produced, the type of data that are being produced, and again, the issue with the storage, the issue with the actual software tools. I mean, you have to remember that, all of these require enormous amount of expertise, enormous amount of resources, different types of data. And I think that is where the NCI CRDC and specifically the CGC and the 3 other cloud pilots, but specifically CGC, makes a huge impact on being able to provide and democratize some of this work. And as you already know, like I said before, that there was a 2-day all hands CRDC meeting, which was really fantastic, and I think some of the cloud resources were also mentioned there. But in a nutshell, I think I want to just briefly say that the CRDC, of course, is a fantastic platform where there is a lot of opportunity for data sharing, data analysis, and data integration. And of course, there are petabytes of data already available, so there's no need for you to download or upload any of this. And of course, this is all done through the different cloud resources, and the FireCloud, the ISB, and of course, the 7 Bridges, which is part of the CGC, or the CGC is part of 7 Bridges. And I think we selected specifically to work with the CGC on the CRDC, and why we picked that is specifically because there are access to, of course, different data types that we already mentioned. There is the TCGA, the CPTAC, and I don't have to tell you guys, some of you guys already know. If not, it's a great idea to go to the CGC website and identified enormous amount of data types that are available there. And of course, they also have a great platform in terms of the training. I've actually looked at some of this myself just recently. And I think this is an older slide. So over 700 different analytical tools and workflows within the CGC platform that can be utilized by non-bioinformatician. And again, this requires very little to no coding. And if you're, of course, a bioinformatics savvy, you do have that choice of working with the code and the command line. But most people that I know, our collaborators are biologists, basic scientists, physicians, they really don't want to deal with that. They just want to go and point and click. So with that in mind, again, having the CGC, here is a couple of, again, anecdotal example of how, if you show this to, again, a non-bioinformatician, and that includes myself, this is a simple screenshot of a RNA-seq pipeline that we are using in our BioWolf HPC within the NIH, and then this is just a small portion of it. So, this is quite overwhelming for, again, people who are not familiar with the coding. Now, in the CGC and the CRDC, you have the ability now to convert this actually from a code into a point click by simply generating this really nice workflow. And again, while I'm not going to go into how this workflow is obviously established, because again, the CGC has a fantastic tutorial for how and to access and how to use them. But here you can see that you have a lot of input data and flexibility that you can go ahead and put in and click in one button or one click. You can have potentially a QC for a lot of the stuff that requires a great deal of coding. This is done for RNA-seq, obviously, and again, RNA-seq has been one of the most widely used algorithms and and data analysis. And you can go ahead and look at the quality of your base pairs. You can look at the actual GC content per base, and you can also do fancy stuff like PCA and also MA plots and volcano plots. So again, this is a really perfect way of, again, using some of the already well-established RNA-seq or potentially mutation analysis or other types of ChIP-seq. These are the most widely used algorithms or data analysis, and they're all available within the CRDC/CGC. And again, they're very easily can be used just by point and click. So again, I think this is, again, these are the low-hanging fruit type of things. And what I wanted to now sort of switch gear a little bit and tell you a little bit about some of the integrative data analysis that we have done with the CGC and why. The rationale for doing the obviously integrative data analysis is that in the past there has always been a notion of reducing the complexity of the data. Meaning that there was a reductionist approach where you start with the sample and then you basically go down all the way to DNA and then focus on one specific data type, whether it was DNA, RNA, or protein. But I think that, as I mentioned before this in my last talk, if you guys were there in the CRDC, the paradigm is shifting in a way where we're now reaching a point where we want to actually be very inclusive and do integrative data analysis. Rather than looking at only one data flavor, we want to actually do now DNA, protein, pathway network, and potentially even looking into the microenvironment of the tumors. And why do we really worry about considering doing integrative data analysis? Because as we all know, that cancer is really not a single gene disease. And we also know that the heterogeneity of the cancer speaks for itself, that there's so many potential errors or potential mutation or potential abnormality within the gene-gene interaction or potential pathway networks that could be causing the development, progression, or the reoccurrence or potentially resistant to treatment within the cancer. So we think that again, the paradigm is shifting, that a lot of people are now looking into potentially being into doing integrative analysis. So with that in mind, again, I think the community is realizing this, and they're developing fantastic tools. And there are a lot of tools out there, but this is the one that we have used in a couple of our projects with our collaborators, which I think that is really a fantastic tool. And also, if I will show you in a minute the actual number of codes that it takes to do this. But let me just tell you a little bit about this algorithm that was published in 2019. And it's known as multi-omic gene set analysis, MOGSA for short. And what they're doing here is that they are adding different flavor of the data. You have copy number variation, you have messenger RNA, you have protein, global protein expression, as well as phosphoprotein. Now, the algorithm called MOGSA, it actually accesses all these data types and then trying to run this analysis using different data sets, databases. Here is listed the CAG, the Reactome, and the MSeq databases. But you can have a database of choice. We actually like to use the Hallmark because it's really well established among the oncological research fields. So once you have these different data flavors and these databases, which obviously contains the pathway and network, then you generate a really nice sort of a heat map. And the heat map, basically, if I can just go over this very quickly, so the columns basically represents the samples, and the rows are representing the pathway, and you actually assign a score. If it is shown in red, that means that it has a very high activity score. If it's blue, that means it has a low activity score. If, of course, it's the yellow, it has really, in between. So we're interested, for example, in this very high activity score right here. And this is specific pathway for this specific sample seem to be very highly, very high activity score for that specific pathway. The question is where that data is coming from. This score is obviously, there's a lot of contribution from copy number variation, from messenger RNA, from protein, and from phosphoprotein. The question is which data type really contribute to this actual activity of this pathway. And now we know, using this algorithm, that it is the messenger RNA that is shown in green, as well as the phosphoprotein. Now, the contribution of these two data types is what gave rise to this high score activity for this specific pathway for this sample. And we can drill down even deeper, and not only knowing that this comes from messenger RNA and from phosphoprotein, but you can look and see which biomolecule, which gene, which actually phosphate, or which site that is responsible for this potential activity, score activity. So again, this really not only integrated the actual analysis, but also it sort of then deconvolute and trying to identify the actual culprit of what really gave rise to that specific, well, what contribute to that potential score. So using this, again, here is the actual code for doing the MOGSA. As you can imagine, this will keep me up at night if I have to really worry about writing or reading this. So, and just think of this being actually the code, and you basically, as the CGC and a couple of my team members have actually done a great job of basically take an image of this code and upload it into either SVG Docker, or if it's a tool of or algorithm of interest that already is in the SVG Docker repository, you use the visual editor just to simply now, and again, I'm not going to go into detail because I think it's something that you don't have to worry about that, because I'm sure the CGC people would be happy to help you with that. So you basically get the image and create this workflow. And once you actually have this workflow, it is just a matter of point and click. So you can imagine going from this into something like this. But just to see, give you a little bit of information. So you have, for example, let's say messenger RNA data from the TCGA. You can identify those. You can also look at the CPTAC protein. And then, of course, if there are other copy number variation of phosphoprotein, you can also bring those data together, search them on the CGC website, identify them. And there's a really cool I would say a function within the CGC that is known as intersect. When you put in your sample and your potential, when you're filtering for the sample, we're in this case looking at the TCGA and CPTAC. Let's say you're looking for RNA and specifically you're interested in breast cancer. There are a lot of samples there, but if you want to overlap the CPTAC with the TCGA, let's say if you're interested in images from TCIA, to put all that data together, CGC has done a really good job of potentially, getting this into a platform which is a lot easier. For example, you now use the intersect to identify all the samples that are present in TCGA, CPTAC, again, for RNA-seq, for protein, for copy number variation, whichever data you put in, specifically for breast cancer. Now we changed from 11,000 samples to only 335. So this really cut down the amount of unnecessary work and also the type of data that is required for you to do your analysis. Now, the actual process of generating the workflow is simply you put in obviously your data, which I just showed you some of the actual messenger RNA, and I think I list some of them right here. So you have your obviously subtop files. These are the clinical phenotypes, which we'll mention in a minute or two. And then you have the phosphoprotein, the global protein, the messenger RNA, the copy number variation, and then of course here is the databases. So once you have all this, they go through some decompression. There has to be some decompressing and some renaming. It's actually called basically process. And once again, based on my conversation with the CGC folks and also my own team members, that you don't have to worry about a lot of these. Once you identify the data that you're interested, you click and the data get decompressed and processed and renamed. And that will take you to, for example, in the case of the MOGSA, to the processed data, the prepared data. Once you have the prepared data, now you have the ability to merge all the data that you decompressed and process. And of course, the processing takes some normalization. In the case of phosphoprotein, it may require some degree of imputation. In the case of RNA-seq, it may require some other types of pre-processing. So once that's all ready to go, of course, the subtypes are, whether something is a reoccurrence, age, race, or potentially if there's treatments. So you can have those clinical attributes. And the databases, of course, are what I mentioned before. We usually use CAG, Reactome, and Hallmark. And so then you have selected these 3 inputs, and all you do is now click on the actual step 3 of the MOGSA, and that generates the pathway network analysis in the main directory, and that will literally spit out a lot of information where it can give you some normalization plots, which I actually did not save here. It can give you potentially some QC, but the final thing that I think my interest is, of course, most, again, clinician and most basic biologists are interested is the final product of course, once the normalization works, once the actual QC is passed, what is the final visualized data? And this is, again, a bit convoluted, and I'm going to go ahead and this is unpublished data, so I have actually masked a lot of the information from this. But here, what you're looking at is the 3 subtype of a specific cancer. Now, these cancer types, the yellow, the green, and the blue, basically went through what I just mentioned to you for the CPTAC, for the TCGA, and with a couple of different data flavors, with the RNA, with the protein, and phosphoprotein. So as you can see, these are, again, samples, the column, and the rows are the pathways. As you can see, I have here pathway names. I removed them. You have pathway 1, pathway 1, pathway all the way to, I don't know why I skipped 2, but from 1 to 8, and then from 9 to 13, and from 14 to 19. As you can see, this sort of a bird's eye view of this actual heat map shows that a really nice pattern is emerging. A pattern is emerging for the yellow subtype and the green subtype and the black subtype of cancer. And this is critical because what this is telling us that these specific pathways are potentially activated in this subtype that are not activated in the green one or in the black one. Or on the contrary, these, the pathway number 9 to number 13 have a high score activity, potentially in the green subtype that is not found in the black or in the yellow. And of course, same thing true for the black one. Not only that, they tell you that actually pathways are, have a high score activity, but now, as I mentioned in my previous slide, you can now drill down and find out what is the contribution of each one of these RNA, protein, or phosphoprotein. What is driving this actual score? As you can see here, in the case of the yellow, the majority of the score comes from the phosphoprotein and some from the actual global protein. RNA does not have a lot of impact on this subtype. In the green subtype, you see that is somewhat a little bit different. It is the global protein that has the most impact on producing this high activity score, where the phosphoprotein has a limited, but again, RNA doesn't have a whole lot. Same thing with the black subtype. The black subtype shows that, again, the global protein and the protein to some extent, and maybe the RNA, has some degree of influence or contribution to this pathway activation. And again, as I mentioned before, since cancer is an actual pathway network disease, a combination of many different genes and pathways, I think this literally opens the door now to potentially look at some of the pathways activity for this specific subtype, and then correlate this to potentially survival, reoccurrence, resistance, and there are plenty of things that one can then do to actually identify the correlation of the significance of these pathways to why they are specific to these subtypes. Now, this is the work that's already been done, and if I'm not mistaken, this is already in production, so you can go and put your data and do your MOGSA analysis. But I definitely want to go ahead and plug in very quickly incredibly important tool that was developed within the CGBB and within our branch. It's called the Omic Circus. It is a visualization tool. It is an absolutely fantastic tool, and I'm sorry I'm bragging about this because a lot of people that use this, they love it because you can see, I think this is an older slide again, about 37,000 downloads since 2013. It is written in R, and if you think that the MOGSA was actually complicated, looking at the code for this are pages and pages, if I'm not mistaken. And right now, CGC is working with my team right now as we speak, trying to actually develop, again, a point-and-click workflow within the CRDC, within the CGC, that can be used to visualize a lot of the data. I'm just going to go very quickly over kind of capability that this have actually. So the very outer layer is simply the actual chromosome, and then there are incredible flexibility here where you can put in then the actual gene. The little heat map that you're seeing here shows the expression of different gene here. And then you can also have potentially copy number variations. You can have fusion, as you showed, whether it's intrafusion or interfusion within the chromosome or within the specific these specifically show, for example, this chromosome is fused with this chromosome. And then also there are actual statistical correlation for each one of these findings. And these are the, again, each one of these tracks can be labeled differently. It can be actually, put in whether it's expression, copy number, mutations. So the sky's the limit in terms of the amount of the type of data that you want to visualize. You can also have a zoom, function within this, at least in the R, and I'm hoping that it will happen in the point and click as well, where you can zoom in and again see the actual samples that you're interested in, or the actual region that you might be interested in terms of whether something is amplified or something is deleted or something is highly expressed versus being involved in fusion or not. So I think with that, if I'm not mistaken, I know I might have gone a little bit fast, but this is all that I have, and I definitely want to thank the 7 Bridges, the CGC folks, specifically Dave Robinson and Manisha, Ray, and of course Zelia, for doing an amazing job for providing the resources and providing the logistics to get all this work done. And of course, my own team members, specifically Trina and Japan was the main person who actually did the work, and then Ying and Chenrong and Chonhua. The Omics Circa is the tool is developed by Ying Hu and with the help of Chonhua and the rest of the team. So with that, I think I will go ahead and stop, and we'll be happy to answer any question. Stop.
Thank you so much, Daoud. That was a great talk. It doesn't seem like we have any questions in the chat, but I do have a question for you.
Okay.
Maybe for your team. How much work was it to build the CGC workflow, and how much help did you need from the 7 Bridges team? Right.
I think most likely Japan is the right person to actually answer this question, but I can tell you from a high level communication that I've had with the team and with the CGC, it was, at the beginning, it was a bit of a heavy lift just because this was fairly new. But once we got the hang of it, it was quite quick. And I think, again, Dave Robinson had been quite accommodating, and some of the other team member within his group. So I would say once we got to know what we were doing in some of the code that were provided for the CGC folks, then it was a breeze, I would say. And I got to be very honest, I just had a meeting with the folks for the Omics Circus. I am blown away how fast and how incredibly good that looks. I don't want to jinx it because, it involves all kinds of technical stuff that is above my pay grade. So I don't want to, but it was really, really very impressive how fast they have been able to generate the Omics Circus. And the same, of course, with the CGC. I think it is Japan. I don't know if you're online, but if you want to chime in, but for the most part, I think it was an easy lift.
Yes, I should say David Robinson was really, really helpful. He led me hand in hand to go along so far. So at the beginning, I don't know what to do, but he was so patient and so helpful and led me all the way through. So, and right now I cannot say I'm already an expert, but I can work on my own and implement some useful tools. So,
and I think I don't want to, again, since this is something that is part of CRDC, it's part of the NCI,
right.
and I think it is of critical importance to let people know that there's a lot of training available. And,
right.
And again, if someone is interested, if the training happens through the CGC folks, or if not, we will be happy to specifically train on the actual MOGSA or when the Omics Circa tool come to fruition. And again, that is the #1 reason why I agreed to do this, because this is a big part of the CRDC, and I know that CRDC is providing a huge service to the NCI and to the outside of the NCI community for data analysis and data sharing. Michael, so how much control does the user have over setting program parameters? Can you run individual steps in workflow? I think I can answer this, but then I will probably again ask Japin to get into the technicals of it. Yes, you can do individual modules or individual steps in the workflow. As a matter of fact, I remember you can pull one out and then I remember when Manisha were doing her breakout session yesterday. It was really cool how she was pulling one workflow into another one and trying to combine and trying to remove them. As far as the flexibility of control, Japin, can you quickly, how much control does the user have?
Okay, yeah, but before that, I should also mention, trainers, so all the backend script are provided by trainer, which as you can see from the Daoud's talk. So that's how much enormous amount of work of that. As for the flexibility, so if you want to develop your tool, so I think, so this tool is pretty much very flexible and easy. So as you see, so the tools are breaking into very small one. Each tool finish one task. So if you want to compose your own workflow, so you can basically drag and drop each individual tool and complete your own workflow. I think it's Steven Bridge did a very good job here. So of course there's some learning curve in the beginning, but once you get a hang of it, it should be pretty easy. So save a lot of work. You don't have to deal with backend code, yet it's very flexible for you to manipulate and do your own workflow.
Okay, great. So Mingyi has a question about, I think this is a great question, Ming, because prior to proteomics, I think obviously we had no choice to sort of rely a lot of the data analysis on our RNA-seq, whether it was on the chip array or on the microarrays or on RNA-seq. But it's my experience now, I could be wrong, that we're finding that in a lot of the projects if the proteomic is present in comparison to the RNA, the proteomics seem to be more informative, or it seems to be more, for lack of a better word, it is more significant in certain cases than the RNA. That, at least that is my experience in a few of the projects that we have been involved that has both protein and RNA. So in terms of, do we actually have different weights assigned for these different data types? My answer is yes, but I will actually either get Chin wrong or Trina. If one of you guys can please chime in, because I don't want to give the wrong answer, because the technical part of it, you guys are experts.
Actually, we assign the same weight for both, like protein and the RNA. In some occasions, we see RNA actually contribute more, but as Daoud just said, most of the cases we see like if the RNA and the protein, we see protein contribution is like more than RNA. But we also see the opposite way, but in most of the cases is protein contribution higher.
And I'm going to go on a limb here and put my, sort of professor hat here, that looking back into, the cell, obviously, and the cancer itself, the majority of the function happens within the protein. Obviously, the RNA is of critically important, but, so having a modification, the RNA may not change much, but if you have a phosphorylation or dephosphorylation of protein, it can really have quite a bit of an impact. And a lot of the, again, some of the oncogenes or some of the oncogenes or oncoproteins, such as, again, VEGF and EGF and EGFRs and all these potential receptors that are either, altered or abnormal in a lot of the cancer, that's an indication that a lot of the treatments are based on, for example, actual protein modifications. So I think that kind of makes me feel comfortable to say that protein may have more impact than the RNA, but of course, I'm making a generalized statement. But going back, I think protein has a huge impact on the fate of the cell. And that's probably why we're seeing, as we are getting more mature and not learning, but also understanding proteomics, because, you know, again, 15 years ago, we didn't really have a good understanding of proteomics, but now with the new technology, with the new computing resources, the proteomics is, of course, coming to fruition. I hope that answers your question, Ming.
Yeah, it does look like it. So I actually had a couple of follow-up questions and comments.
Yep.
For the developer question from Michael. If there's something specific that you're thinking about, we do have office hours that you can attend, and we're happy to help you. In addition to that, we allow to do what the developer lets you do. So if the more control the developer wants to give you, then the more control you have about from the app. In addition, I wanted to ask Daoud if it would be possible to get a quick overview of the CRDC. Maybe a lot of people on the call are not as familiar with the Cancer Research Data Commons. And so if we could get like a couple of minutes of what it is and how it's helping cancer research.
Yes, absolutely. I think, let me see if I can just quickly, there's a really fantastic video that the CRDC folks have put together, and it is, in a nutshell, it gives you a really awesome overview. But in a short couple of sentences, CRDC is the Cancer Research Data Common. It is a sort of a, you know, it's a huge cloud, as is shown right here. I don't know if you guys can see my my slides still. So here is the actual CRDC, and within the CRDC, there are enormous amount of small clouds. In these, well, when I say enormous amount, there's quite a few of them. And each one of these clouds has different capabilities. For example, the cloud resources, as we're discussing, you know, the CGC, the 5 clouds, and the ISB has the capability of actually doing analysis. Some of these cloud, of course, contains the PDC, which is the proteomic data common. And there's also IDC, which is the imaging data common. And there are other, you know, other kind of information such as clinical attributes. So the goal here is to, again, I know that people are using this word now too much, democratize the data within the NCI, and within the cancer community. And the way to do that is, so you have the TCGA, you have the PDC, you have the target, and there's going to be a lot more data, you know, pulling in to this CRDC. The goal here is, rather than you going to actually download the data and cost, you know, basically a huge amount of resource and a huge amount of time, you can go to CRDC, identify the type of data that you're looking for, you know, parse that data, put it into one of these clouds, and do your analysis. And so that's just for a simple analysis. But then if you want to do something like we talked about, integrative analysis, you can bring in, for example, data from the imaging group from the IDC. You can bring, you know, again, a protein from the PDC, and copy number, messenger RNA. And there's, again, I can't really go into the detail. There are over 11,000 or potentially even more. I think there's 15,000 different data types. There's it's a huge platform that contains a lot of different, again, protein, RNA, mutations, the raw data, level 3. So it's probably requires an entire hour to go over the whole entire thing. But I think that CRDC all hands meeting that we had yesterday and the day before yesterday provided a really fantastic overview, and that will be available. And I will go ahead and share that link with you guys. So if anybody's interested in more detail, they can go ahead and see that. But then again, in a nutshell, CRDC is basically a one-stop shop place where you can go and do data sharing, data analysis, and hopefully data management. I hope that answers the question.
It does. It's perfect. I also dropped in the chat the link to the datacommons.cancer.gov, which will bring our.
They have a really beautiful video that I think people will enjoy watching it.
Yeah, the video is in that page, so it's
great.
a really nice video. We do have one more question, more biology focused. Could the overall stability of protein versus mRNA in the cell contribute to the lower impact of mRNA in the subtype of cancer, I believe?
Absolutely. I think that's a very important point you're making, Manisha, because of course, mRNA and RNA in general is very, very prone to degradation, where protein is a lot more stable. And there are actually studies that have shown that you may not see the difference in RNA, but you do see in the protein, and that is where they're aiming that this has to have something to do with the stability. And also, you have to remember the manipulation that is going on with some of these work could potentially cause. I remember reading something a long time ago that from the moment that you actually put in your mRNA into the experiment and the time that you're getting it out for the actual analysis, final analysis, you only get about 40% of the data that you put in. I know it's quite scary to think that way, but because of all the manipulation, even though with some of the fantastic tools and technology that we have, still RNA is incredibly labile, and it really has issue in terms of degradation. So stability makes a huge impact. Yes.
Thank you so much. Do we have any more questions? Okay. So thank you so much for attending our CGC webinar. I just put in the chat the link to our office hours if there's more questions that you may have that we didn't answer here. And if you have any questions about the CRDC, we're happy to help and send you to the right person as well. So thanks again, and feel free to contact us if you have any questions. This is recorded and will be available on our website as well.
Great. Thank you all
so
for joining. Appreciate the opportunity.
Thank you so much, Daoud. It was great. Thank you so much.
Bye-bye now.